Hello students, in this video we are going to learn about microbes in human welfare. First we can see about chapter outline. Microbes in household products, microbes in industrial products, microbes in sewage treatment and energy generation, microbes in the production of biogas, microbes as biocontrol agents and biofertilizers, and bioremediation. So these are the topics we are going to learn in this lesson. So in this video we are going to learn about microbes in household products and microbes in industrial products. Okay, introduction. So the major components of biological system of earth are microbes. Okay, here the microbes such as bacteria, fungi, protozoa, algae, viruses, viroids and prions. So these are all the major component of biological system in earth. Okay, and several microbes are beneficial. Okay, that means they produce many beneficial products and to the human welfare. Okay, that is contributed to the human welfare. Okay, so these microbes present everywhere. Okay, and we can grow bacteria and fungi in a nutritive medium in the laboratory. So at that time it form colonies. Okay, these colonies can be easily visible to our eyes. Okay, we cannot see the single cells of bacteria or fungi, but if it is found in colonies, it is easily visible to us. So first we are going to see about microbes in household products. So in our everyday life, the microbes and their products are used for the preparation of idli, dosa, cheese, curd, yogurt, tough bread, vinegar, etc. So here now we are going to see this one by one. First one is curd. We are going to see about curd. LAB, that is lactic acid bacteria. So what are the lactic acid bacteria means? Lactobacillus acidophilus. Okay. So the lactic acid bacteria are Lactobacillus acidophilus, Lactobacillus lactis and Streptococcus lactis. So these are all the lactic acid bacteria. So these bacteria are probiotics. So what do you mean by probiotics? They are beneficial bacteria. So what is the main function of these bacteria are? They check the growth of pathogens in our stomach and other parts of digestive tract. Okay, so mainly these bacteria grow in milk and convert the milk into curd. Okay, how it will convert the milk into curd means by digesting the milk protein casein. Okay, so when compared to milk, curd is more nutritious because it contains number of organic acids and vitamins. Okay, then ergot. So ergot, it is produced by bacterial fermentation of milk. Okay, so with the help of bacteria, the milk gets fermented. So at that time, it produces lactic acid as the byproduct. Okay, so the microbes involved in the formation of ergot are Streptococcus thermophilus and Lactobacillus bulgaricus. Okay, so what it will do means it will coagulate the milk protein. Okay, and also it convert the milk sugar, that is lactose. Lactose is a milk sugar. Convert the milk sugar lactose into lactic acid. Okay, and the flavor of the ergot is due to formation of acetaldehyde. Okay, cheese. So cheese is a dairy product. Okay, and it has a wide range of flavor and textures. Okay, how it is formed means by coagulation of milk protein casein. So next we are going to see about method of cheese production. So first they will acidify the milk. Then they add an enzyme rennet. So this rennet coagulate the milk protein casein. Then the solid is separated. So after coagulation, the milk is separated into solid and a liquid portion. Okay, that solid portion is separated and pressed into cheese. What are the bacteria involved in the formation of cheese are Lactococcus, Lactobacillus and Streptococcus. These are the bacteria involved in cheese making. So next we are going to see about paneer. Okay, so paneer is made by curdling of milk with lemon juice or with the help of vinegar or other edible acids. So if you see the Swiss cheese, so you can see many large holes, that is due to the production of large amount of carbon dioxide by the bacterium. So here for the formation of paneer, So what type of microbes is used means it's a bacteria Propionobacterium cerumani. It is used to make paneer from milk. Okay, next tough. The tough for idli and dosa is how it is made means it is fermented by bacteria Leuconostoc mesenteroids. So this bacteria ferment the tough of idlis and dosas. Okay, and the tough for bread, how it is fermented means with the help of Saccharomyces cerevisiae. And what will happen at the time of fermentation means this yeast, Saccharomyces cerevisiae is a yeast. Okay, it will ferment the sugar, glucose. Okay, it will ferment the glucose present in the tough. Okay, thereby it forms ethyl alcohol and carbon dioxide. So that leads to leavening of tough. If you bake this leavened tough, okay, the carbon dioxide and ethyl alcohol evaporate. Thus, it makes the bread porous and soft. Okay, next is single cell protein. So, single cell protein is actually a edible unicellular organisms like spirulina. How they extract the protein means the protein is extracted from pure or mixed culture of algae, fungi and bacteria. Okay, and this protein is used as substitute for protein rich foods. Okay, and this single cell protein is suitable for human consumption as well as it is used as an animal feed. So next we are going to see about microbes in industrial products. Okay, so microbes are used to synthesize number of products like beverages, okay, antibiotics, organic acids, amino acids, vitamins, biofuels, single cell protein, enzymes, steroids, vaccines, pharmaceutical drugs. Okay, these are all produced with the help of microbes. Okay, for the large scale production, so we need to grow a large amount of microbes. Okay, so for that we need large vessel that is called as fermenters. So what do you mean by fermenter? It is also called as bioreactor. Okay, actually it is a closed vessel with adequate arrangement like aeration. So often we have to give aeration for the growing microbes. Then it should be constantly agitate. Okay, then temperature, pH control, drain or overflow vent. So all should be there in the fermenter. So what is the use of this vent means? This vent is used to remove the waste biomass of the cultured microbes along with the products. Next we are going to see about antibiotic production. Okay, so antibiotic, what is antibiotic? It is nothing but chemical substance which is produced by microorganisms and which can kill or retards the growth of other disease causing microbes, that is pathogen, even it is present in low concentration. So antibiotics are mostly used to treat diseases like plague, meningitis, diphtheria, syphilis, leprosy, tuberculosis, etc. Okay, so here we are going to see some of the antibiotics. First we are going to see about streptomycin. So it was discovered by Selman Waxman. Okay, he was the first scientist who used the term antibiotic in 1943. Okay, but the first antibiotic was discovered by Alexander Fleming. That is, the antibiotic name is penicillin. In 1926, he discovered this antibiotic. So how he discovered this antibiotic means, so when he was worked with Staphylococci bacteria, okay, he found a green mold growing in one of the unwashed plate. Okay, so around that green mold, he couldn't see any growth of Staphylococci bacteria. Okay, so then he examined the plate. There he found out a chemical substance produced by the green mold. Okay, then he named that chemical substance as penicillin. Okay, so penicillin is produced by fungi Penicillium notatum or Penicillium chrysogenum. Okay, these are the two species of fungi which produces penicillin. Okay, so it is a bactericidal in action. That means it has the ability to kill the bacteria. So how it will kill the bacteria means by inhibiting the synthesis of bacterial cell wall. It is a broad spectrum antibiotic. Okay, so it is referred as queen of drugs. The next antibiotic is tetracycline, which is one of the broad spectrum antibiotic. Okay, it is a bacteriostatic antibiotic. That means it has the ability to control the growth of bacteria. How it will do means by inhibiting the bacterial protein synthesis. So you know that protein is needed for it, for the growth of any organism. Okay, so it will inhibit the growth of bacteria by inhibiting the protein synthesis of bacteria. Okay, next, chlorotetracycline. Okay, it is the first antibiotic in this group of antibiotic, and it is isolated from the culture Streptomyces aureofacium. Okay, and next one is streptomycin. So streptomycin is also one of the broad spectrum antibiotic, and it is isolated from actinomycetes and Streptomyces griseus. Okay, and it is a bactericidal. Bactericidal means it can kill the bacteria. Okay, so both gram positive and gram negative bacteria, especially it controls the growth of Mycobacterium tuberculosis. So it is used as a drug for tuberculosis. Okay, then other type of antibiotics are erythromycin, chloromycetin, grisofulvin, neomycin, kennamycin, bacitracin. These are the other type of antibiotic which is extracted from the microbes, isolated from the microbes. Next we are going to see about antibiotic resistance. So what do you mean by antibiotic resistant means? Usually it occurs when bacteria develop the ability to defeat the drug designed to kill or inhibit their growth. Okay, that condition is called as antibiotic resistance. So how it is happening means, so often if you use the antibiotic, unnecessarily if you use the antibiotic, that will develop the resistant to the bacteria. Okay, so thereby the bacteria get resistant against that antibiotic. Okay, so you have to use the antibiotic when it is prescribed by the certified health professional. So how can we prevent this antibiotic resistant of bacteria means by using narrow spectrum antibiotic instead of using broad spectrum antibiotic. If you use the narrow spectrum antibiotic, so it will effectively and accurately target the specific pathogen and it kills that microorganisms. Okay, the disease causing organisms. So thereby we can prevent the resistance of bacteria to the antibiotic. Okay, and what do you mean by superbug means? It is a term used to describe the strains of bacteria that are resistant to the majority of antibiotics commonly used today. So next we are going to see about fermented beverages. So especially yeast is used for production of beverages like wine, beer, whiskey, brandy and rum. Okay, so among these beverages, wine is the oldest alcoholic beverages known. Okay, how it is formed means by the fermentation of fruit juice, like with the help of yeast. Zymology. So what do you mean by zymology? So it is a applied science, deals with the biochemical process of fermentation and its practical use. So usually in the beverage industries, so what type of microorganism is used means Saccharomyces cerevisiae. Okay, so this species of yeast is called as brewer's yeast, and usually this yeast ferment the malted cereals and fruit juices, thereby it produces alcoholic beverages. Wine and beer are produced without distillation. These are the beverages produced without distillation, and whiskey and brandy and rum are obtained by fermentation and distillation. They are distillated beverages. Next, enology. What do you mean by enology? It is a science and study of wine and winemaking. Usually wine is made from fermentation of grape juice. Based on that, the wine is divided into two types, that is red wine and white wine. So red wine is made from black grapes. It is used along with their skin and stem. Okay, it is produced along with the skin and stem. In the case of white wine, it is produced from the juice. Either white or red grapes are used here without skin and stem. This is the difference between red and white wine. Next is beer. It is produced from fermented barley malt. Okay, fermented barley malt grain. Okay, with the help of the microbe Saccharomyces aspergillosis or by using Saccharomyces cerevisiae. Next, rum. So rum is made from fermented sugarcane or molasses, or it can be prepared by directly from sugarcane juice with the help of the yeast, that is Saccharomyces cerevisiae. Next, whiskey. Actually, it is a distilled alcoholic beverages. Whiskey is the distilled alcoholic beverage which is made from fermented grain mash with the help of Saccharomyces cerevisiae. So next, padanir. So padanir, it is obtained from fermented sap of palms and coconut trees. Actually, it is a refreshing drink. By boiling this, we can produce jaggery and palm sugar. Okay. So when it is left undisturbed for few hours, it is converted into fermented form that is called as toddy. In Tamil, we can call it as kal. So this toddy is a beverage which contains 4% alcohol. Okay, and if it is left for 24 hours, so after 24 hours, it becomes unpalatable. That means we cannot able to consume it. Okay, that is called as unpalatable, and thereby we can use that for producing vinegar. So next we are going to see about ethanol. So Saccharomyces cerevisiae, this yeast only a major producer of ethanol, and ethanol is used for industrial, laboratory and fuel purpose. So ethanol is referred as industrial alcohol, and the bacteria Pseudomonas mobilis and Sarcina ventriculae. It is also used to produce ethanol. Okay, and the principal substrate for the production of ethanol is molasses or corn. Along with that, they will add potatoes and wood waste. Okay, the process of ethanol production starts by milling of feed stock, that means waste wood. First they will break that waste wood. Thereby, after that, they will add dilute fungal enzyme, that is amylase. So this enzyme is extracted from the fungi Aspergillus. So this will digest the starch into fermentable sugar. So starch is a polysaccharide. It is converted into sugar. Then yeast is added to the mixture. That it converts the sugar into ethanol. And after the production of ethanol, it gets distillated. It is distillated to get 96% concentrated ethanol. See this equation: glucose, when it is fermented with the help of yeast, will produce ethanol by the liberation of carbon dioxide. So next we are going to see about chemicals, enzymes and other bioactive molecules. So microbes, not only it is used to produce many industrial products, but also it is used to produce many types of chemicals, enzymes and bioactive molecules. So here we are going to see about the microbes which are used to produce many organic acids. Okay, here Aspergillus niger. So this fungi is used to produce citric acid. Okay, then Acetobacter is used to produce acetic acid. Then Rhizopus oryzae is used to produce fumaric acid, and Clostridium butyricum is used to produce butyric acid. Then Lactobacillus is used to produce lactic acid. So next we are going to see about the enzymes. Okay, so with the help of microbes we can also produce this enzyme. So first one is lipase. So this enzyme is used in detergent formulation. Thereby it is used to remove oily stains in the laundry. Then pectinase, protease and cellulase. So this is used to clarify bottled juice. Then rennet. So this rennet is used to separate milk into solid curd for cheese making. Then streptokinase. So streptokinase is produced by streptococcus. Here what they will do means genetically engineered streptococci are used as clot buster. Okay, so that means it will remove the clots from the blood vessels of a patient who had undergone myocardial infarction. Next one is cyclosporin A. So cyclosporin A is acting as immunosuppressant. That means it will suppress the immune system. So because of that, it can be used for organ transplantation. So at the time of organ transplantation, they will keep the healthy organ of some other person. So in order to accepting of that organ, so the patient should be given the immunosuppressant drug. And this cyclosporin is produced by the fungi Trichoderma polysporum. Also it is used in, it possesses the property of anti-inflammation. Okay, anti-inflammatory, antifungal and parasitic property. So it can be used as an anti-inflammatory drug, anti-fungal drug, and also anti-parasitic drug. Next one is statins. So statins are produced by yeast Monascus purpureus. So mainly it will lower the blood cholesterol levels. How it will lower the blood cholesterol level means this statins compete with the enzyme which is responsible for the synthesis of cholesterol. Okay, that way it will inhibit the action of the enzyme which will produce the cholesterol. Thus it will lower the blood cholesterol levels. Okay, next recombinant human insulin. So recombinant human insulin is produced in large scale by using E. coli, genetically engineered E. coli and Saccharomyces cerevisiae. So thereby the insulin is used for therapeutic purpose in human beings. Okay students, I think you would have understood the concept. In the next video we can see about the remaining topics. Okay, thank you students.